Now, this is something that we talk about in the lab as well. These are respiratory volumes. These are volumes that we can use to assess the status of your respiratory system. Things like tidal volume. Remember, I think of the tide coming in and out. So tidal volume is just normal breathing in and breathing out. Okay. This is just normal, quiet breathing, and it's the amount of air that moves into and out of your lungs during normal, quiet breathing. Inspiratory reserve. This inspiratory is the key word here, inspiration. It's the amount of air that you can inhale with maximum effort after tidal volume. So how much more can you breathe in after a quiet inhale? Expiratory reserve is kind of the opposite. Keyword is expiratory. So this is how much air can you exhale with max effort after tidal volume. And then residual volume is just, remember, you never can blow out all of the air from your respiratory system. There is always going to be something left, keeping the bronchial tree inflated. That's the residual volume. So it's the air that's left in your lungs after expiratory reserve. You never blow it all out. Now, we also have combinations of these. Vital capacity is the total amount of air that you can exchange. So that would be tidal volume, normal breath in and out. Inspiratory reserve, how much more can you breathe in? And expiratory reserve, how much more can you blow out? That's all the exchangeable air, okay? And then total lung capacity is everything. So it is tidal volume, inspiratory reserve, expiratory reserve, and it also includes this last one on here, residual volume, everything that's left. Okay, and typically, total lung capacity is somewhere around 6,000. Now, remember, in terms of this residual volume, I didn't mention this, residual volume is around 1,000 for females, and it's around 1,500 for males. So this number here, 1,200 mils for residual volume, that's just sort of an average. This is a great visual from your textbook. So this is showing you right here in that light blue area, that would be your tidal volume, normal breathing in and out. Okay. You can see in this sort of purplish color up here at the top, this is your inspiratory reserve. How much more after tidal volume can you breathe in? Expiratory reserve is in the darker blue. How much more can you blow out after tidal volume? And then residual volume, you can see that that line is not moving down into this green residual volume. This is not exchangeable air. It's what's left in your airway after you've blown out as much as you think you can blow out. There's still some left, okay? And I like this graph because it also shows you the combinations here. So it shows you vital capacity. That's all of the exchangeable air. And it shows total lung capacity, which is everything. So this is a good visual. Now we also have something called anatomic dead space. This does not contribute to gas exchange. This is the air that stays in passageways. So this isn't air that's being used to move into your bloodstream and out of your bloodstream. This is staying in the passageways. So it doesn't contribute to gas exchange. And it's about 150 mils. And then alveolar dead space would be anytime you have air moving into non-functioning alveoli. They're not contributing to gas exchange either. Non-functioning alveoli would be someone, for example, who has something like emphysema. Emphysema causes alveolar walls to break down. So instead of having all these nice little grapes in here, you have this one giant grape. And so it's not doing nearly as good of a job at gas exchange. So how do we measure all of these different air movements and lung function? Is we use something called a spirometer. So this is the instrument we use to measure these volumes and capacities. And a spirometer reading, when you do a spirometry reading, it can be used to distinguish between things like obstructive diseases and restrictive diseases. So what does that mean? An obstructive disease, think of the word obstruction. This is something that is going to cause the resistance in your airway to be high. It's obstructing your airway. It's making it really difficult for you to get air into your lungs. That's an obstructive disease, okay? And so typically what happens, this would be like bronchitis, where your bronchioles are kind of constricted and small, you have mucus in there, you have a lot of resistance. And so when someone has something like bronchitis, they breathe typically like this. They hyperinflate. They have to use a lot of energy to get that air down in there. And they end up hyperinflating their lungs. This actually over time causes their tidal volume to go up because they're so used to hyperinflating, and that's their normal tidal volume, that tidal volume in someone who has an obstructive disease is going to be high. A restrictive disorder, this is going to actually prevent your lungs from expanding fully. So they can't actually expand all the way. And if your lungs can't expand all the way, then your tidal volume is going to be low. So when we say that we can use a spirometer to assess lung function, they can actually use the readings. If your tidal volume is low, then they might say, oh gosh, you've got a restrictive disease. Maybe somebody has sunken chest, or maybe they have scoliosis, so they can't expand their lungs all the way. Or if their tidal volume is really high, they could say, oh gosh, I bet they have asthma or bronchitis, or they have some other restrictive disorder where they're constantly hyperinflating their lungs. So it's a good way to assess some of these disorders. Now, we also have some non-respiratory air movements that don't really fit in with tidal volume and inspiratory and expiratory reserve. These are basically some non-respiratory air movements that can sometimes modify the normal breathing rhythm. So these are sometimes our reflexes. Some of them are voluntary, some are involuntary. So these are things like coughing or sneezing. Obviously, these change your breathing rhythm, right? Before I sneeze, I'll go, like I'll take big breaths in and then I'll sneeze, right? So it'll change your normal breathing rhythm. Laughing and crying, hiccups, those change your breathing rhythm. And yawns. We don't know what causes some of these. Like, for example, we don't actually know what causes a yawn. Some people have suspected that it has to do with blood flow to the brain. You're not getting enough blood flow to the brain, so you yawn, you open everything up, and it helps to increase pressure and drive blood up to the brain. But that doesn't explain then why a yawn is contagious, because when you see someone yawn, you yawn too. When I talk about it, it makes me want to yawn just talking about a yawn. And so that doesn't explain some of the other things that we see with yawns. But all of these, because they're changing our normal breathing rhythm, they really do help to promote the flow of blood and also lymphatic fluid. Okay, so the last part of this lecture that I want to walk you through has to do with gas exchange. So the whole point of respiration is to bring oxygen into the alveoli and then carry that oxygen from the alveoli into your red blood cells, and then carry it to your tissues. The other point is expiration, which is taking a waste like CO2 from your bloodstream, putting it into your alveoli and getting it out of your body. So inhalation and exhalation are driving gas exchange. And so we really, when we talk about gas exchange and we talk about this happening, we're going to kind of follow it from two aspects. One is external respiration. This is happening in the lungs. So this is called external respiration. So this is gas exchange between the lungs and your bloodstream. Internal respiration. This is deep inside your body, and this is the diffusion of gases between your tissues and your bloodstream. Okay, so we're going to kind of talk about it from both of those aspects. We'll start with external respiration. So this is the exchange of oxygen and CO2 across the respiratory membrane. Now, we spent in the first lecture quite a bit of time talking about the respiratory membrane. That's the wall of the alveoli, the basement membrane, and then the wall of the capillary, because that is what the gases have to diffuse across. Okay, so that respiratory membrane. So in terms of external respiration, this is going to be influenced by three different things. One is the thickness and surface area of the respiratory membrane. The thicker that membrane is, the harder it's going to be to drive gases across. Okay, the thinner it is, the easier it is. Another thing that will influence external respiration is partial pressure gradients and gas solubilities. And we'll talk about these in a few minutes. And then the last one is something called ventilation, perfusion, coupling. So we're going to talk about all three of those and how they influence gas exchange at the lungs. So again, the first one is the thickness of the respiratory membrane. So normally it should be anywhere from about 0.5 to 1 micrometer thick, so very thin, but it has a huge surface area. Remember, all these alveoli, if we were to stretch them out, they're about the size of a tennis court. So they're huge if we stretch them out. So it's a huge surface area, 40 times the surface area of your skin. So it's nice and big. We have this huge surface area for gas exchange. What can cause the respiratory membrane to get thicker would be like if your lungs become waterlogged. So have you heard of the condition called dry drowning? Has anybody heard of this? This is very dangerous. This would be like when a child gets submerged underwater. Maybe they're swimming in the pool and someone finds them, grabs them, gets them out. They cough, they sputter, they seem fine. They may play and be fine. And then they put them to bed at night and they end up passing away. That's a condition called dry drowning. So they have seemed fine, but what has happened in the process, those couple of hours since that event, is that the water has stayed in their lungs and it's actually caused gas exchange to become inadequate. And you don't know it. Now, they may get real sleepy and that's why you're putting them to bed, but it is a very dangerous condition. So it's really important if they go underwater and even if you pull them up and they're sputtering, to have them checked out, okay? So that's one thing that can influence the thickness of the respiratory membrane. Another one would be decreasing the surface area. Clearly, the surface area for gas exchange is important. If we have all these teeny tiny alveoli, if we stretch them out and they're the size of a tennis court, that's huge. That's a big deal. So what happens is if you start to lose surface area, like if alveolar walls break down, and instead of having all these tiny alveoli, you have giant alveoli, you've just cut the surface area down a lot. And so that's really going to influence gas exchange. Other things that can influence surface area would be things not just like emphysema, but tumors and mucus and inflammation. So the second thing that will influence external respiration, so gas exchange at the lungs, is partial pressure gradients for these gases. So a partial pressure gradient, typically what we'll find is that it's very steep for oxygen. So the partial pressure gradient is truly the pressure difference between what's in the lungs and what's in the bloodstream. Okay, just like we talked about when you breathe in and out, it has to do with pressure changes between what's in the atmosphere and in your lungs. Now we're talking about the pressure of these gases in the lungs versus your bloodstream, okay? And that's going to drive the flow of these gases. So it's very steep for oxygen. So what we find is that in the blood that's coming up to the lungs, the partial pressure gradient for oxygen, the pressure is about 40 millimeters of mercury per oxygen. And in the alveoli, when you breathe in, the pressure of oxygen in the alveoli is 104. Remember, you always move from a high to a low pressure, okay? So what way would oxygen move? You have a whole lot of it in the alveoli and you have less of it in your bloodstream. So oxygen's going to go from where it's high in the alveoli to where it's low into your bloodstream. Okay, it's going to go from high to low. So this is driving the flow of oxygen into your bloodstream until equilibrium is reached. That's how diffusion works, right? Equilibrium will be reached really quickly. Usually it's in 0.25 seconds. That is about 1/3 of the time that red blood cell is around the lungs. That's important. You want it to be really fast because even if your blood flow goes up 3 times, you're exercising, cardiac output's going up. If your blood flow increases, it's okay because you are able to diffuse that oxygen much faster than at rest. So you're able to continue to diffuse it appropriately when you're exercising. Okay, even if you speed up blood flow. So this image is showing you oxygen movement, or that's the one we're going to be looking at. You can see here, this is the alveolus. So you're looking at the alveolus here in blue. This is where the oxygen would be in here. You breathe in. You can see over here in red, this would be our capillary. You can see the red blood cell in here. Okay. And so remember the pressure gradient, if we're just looking at oxygen, it's 104 in the alveolus when you breathe in, and it's 40 in your red blood cell. So oxygen's going to move where there's more to where there's less, high to low, until equilibrium is reached. So this is how we're driving oxygen into our red blood cell. CO2 is a little different. CO2 does not have nearly as steep of a pressure gradient. What we find is the amount of CO2 in your veins is 45, and CO2 in your alveoli is 40. This is not as steep, but there's still a gradient. So CO2, again, is going to move from high to low. So where you have more is in your bloodstream. Where you have less is in your alveoli. So CO2 is going to go from your bloodstream into your alveoli, and you're going to blow it out of your body. Okay. So even though the gradient's not as steep, it's still going to move with equal amounts with oxygen. It's still going to move just as fast as oxygen. And the reason for that is because CO2 is 20 times more soluble in blood plasma than oxygen is. So it's much more soluble, so it can move much faster than oxygen, even though the pressure gradient's not as steep. And then the third thing that's going to drive external respiration is something called ventilation perfusion coupling. So before we can understand ventilation perfusion coupling, we have to know what those terms mean. So perfusion, if you just say the word perfusion, we've used this term before. Perfusion just means blood flow to an area, right? You're perfusing a tissue. So perfusion is blood flow reaching the alveoli. Ventilation, if you hear the word ventilation, I always think of breathing, right? Ventilation, a ventilator. It's breathing, it's gas. So ventilation is the amount of gas reaching the alveoli. So when we say that we have to couple these two, what we mean is you have to be able to couple blood flow going to the alveoli as well as gas exchange within the alveoli. They have to be matched for efficient gas exchange. So typically what we'll say is that the amount of oxygen or the pressure of oxygen controls perfusion. And again, perfusion is blood flow reaching the alveoli. So oxygen levels will control the blood flow, the perfusion around the area of the lungs by changing the diameter of the arterioles. The arterioles are big, you're going to get more blood flow into the lungs. If the arterioles are constricted, you get less flow into the lungs. And it's oxygen levels that will contribute to that in your body. CO2 levels will control ventilation by changing bronchiole diameter. If the amount of CO2 and your body starts to go up, that's a waste, right? We got to get rid of it. We got to breathe faster. So if CO2 levels start to go up, your bronchioles typically dilate and get bigger so that you can move air faster, and you'll typically breathe faster to help with that gas exchange. Okay, so CO2 levels control ventilation, breathing rate. Oxygen levels control blood flow getting to the lungs, and those have to be coupled to each other. So that's external respiration. That's what's happening here in the lungs. We also have internal respiration, which is gas exchange in the body's tissues. And so it's very similar, but the partial pressure gradients are just reversed. They're just the opposite. So oxygen levels in the tissue are always going to be lower than what's in the bloodstream, right? Because you're taking nice oxygen-rich blood into a tissue. So what's in the tissue is always low. You've already used all the oxygen. So we got to put oxygen in it. So tissue oxygen is always lower than what's in the bloodstream. So that means that oxygen is going to flow from your blood into your tissues. CO2 is a waste. It's what you're making in your tissues. You're going to take that waste from the tissues, put it in your bloodstream, and bring it back to the lungs to blow out. Okay, so from the tissues to the blood. So it's just the opposite when we get to the tissues. So the last thing we're going to talk about, so that was external respiration and internal respiration. The one thing that we're missing here is how are we moving oxygen and CO2? That's called the transport of respiratory gas. So once we go through external respiration and we fill our blood up with oxygen, how are we going to take it out to our tissues? How do we carry that oxygen? And then once we do dump the oxygen in our tissues and our blood picks up CO2, how do we carry that CO2 back to our lungs and blow it out? So that's called the transport of respiratory gases. So we'll talk about oxygen and CO2 transport. They are very different from each other. Oxygen is super easy. Oxygen's carried in your blood. Only 1.5% of oxygen is dissolved in blood plasma, so practically none. The other 98.5% of oxygen is bound to the iron and hemoglobin of your red blood cells. Now remember, one hemoglobin has 4 iron atoms. That means that it can carry 4 oxygen molecules per hemoglobin. And remember, one red blood cell has about 250 million hemoglobin molecules in it. So one red blood cell can carry about 1 billion oxygens, okay? So most of your oxygen is just being carried in red blood cells. Remember, red blood cells are bags of hemoglobin. That's their job. Now, hemoglobin is given different names when oxygen is bound to it. As soon as we load oxygen onto hemoglobin in the lungs, we call it oxyhemoglobin. That hemoglobin is going to move in the red blood cell all the way to your tissue. And when it gets to the tissue, it has to release the oxygen and put it in the tissues. And when that happens, we call it reduced hemoglobin. We also can call it deoxyhemoglobin. That's when it lets go of the oxygen so it can go into the body's tissues. Okay, so hemoglobin's released that oxygen. And so you can see the equation down here. So you can see this is just hemoglobin plus oxygen. Okay, we bind them together and we form that oxyhemoglobin. That's showing you that right there. Okay, and that process, so moving in this direction, that's the lungs. Okay, when we're binding oxygen to hemoglobin, that's happening in the lungs. If we reverse this and we let go of the oxygen from hemoglobin, okay, now you can see it's unbound from hemoglobin. That's what's happening in the tissues. So that's why this equation is showing it forward and backward. The rate of loading and unloading of oxygen, it's always regulated to make sure that you are getting enough oxygen to your cells. So there are things that can influence oxygen loading and unloading. One of them is high temperature. When your body temperature goes up, you're going to increase the release of oxygen from hemoglobin. That makes sense. right? You're working out, you need oxygen to get into your tissues faster. So you're going to increase the rate of release of oxygen from hemoglobin. You're sick, you're running a fever, your body temperature is high, your tissues need to repair all the damage that's been done. And so you're going to release oxygen faster, okay? Anytime you have high hydrogen ions, this is high pH, or sorry, this is low pH, so it becomes more acidic. So anytime your blood pH drops and becomes more acidic, you have more hydrogen ions, you're going to increase oxygen release. Now, one of the things that we did in lab last week is you did that phenol red experiment, right? And you blew into phenol red. What happened when you blew into the phenol red? It changed color, changed to yellow. Why? Phenol red's an indicator solution, right? And it turns yellow in the presence of carbonic acid and acid, right? So CO2, when it's dissolved in a liquid, is an acid. So when you have a lot of extra CO2 in your blood, it's going to drop your blood pH. It becomes more acidic. So your body's going to say, ooh, we need to release oxygen faster and get into our tissues faster. We're going through cell respiration quicker. Okay, so that's going to trigger oxygen to get released faster into your tissues. If you have low oxygen levels in your tissues, you're going to release oxygen faster. Or if you have high carbon dioxide levels, you're going to release oxygen faster. Okay. Now these two go together. Okay. High CO2 levels, CO2 dissolved in a liquid is an acid. So that's going to mean more acidic. Okay. So those two kind of go together. Now, there are some imbalances that are associated with this, things like hypoxia. Hypoxia is when you cannot get enough oxygen into your tissues, and it results in that condition called cyanosis. Do y'all remember that? Cyanosis from AP1? It's right as when your lips turn blue, your fingernails, sometimes the bed of your nails will look blue in color. This is showing you that you do not have enough oxygen, because remember, when your blood is fully oxygenated, it's nice and red. If you don't have enough oxygen, it's more of a purpley blue color. So there's different kinds of hypoxia. Remember, hypo means low oxygen. Anemic hypoxia. This is too few red blood cells. Or maybe you just have abnormal hemoglobin. Ischemic hypoxia. Ischemia is when you impair or block blood flow to an area. You've all experienced this. If your legs ever fallen asleep, maybe you're sitting funny, sitting cross-legged. I can't sit crisscross applesauce. My feet fall asleep in like 2 minutes when I do that. And that's because I'm blocking a major vessel that is supplying my feet. And when that happens, when you block or impair circulation, it causes ischemic hypoxia. You get that tingly numbness feeling in the limb. Histotoxic hypoxia. This is when your cells can't use the oxygen that's present. A lot of times this will happen with metabolic poisons, things like cyanide. You might have enough oxygen present, but you're not able to use it. And then another one is carbon monoxide poisoning. So this is why carbon monoxide is so dangerous, especially you get carbon monoxide from fire. The reason is carbon monoxide has a 200 times greater affinity for hemoglobin than oxygen. So that means if carbon monoxide is present in the atmosphere and you breathe it in, it's going to bind to hemoglobin way before oxygen does. Well, now your hemoglobin is taken up with something and oxygen can't bind. But carbon monoxide does not serve cell respiration, right? Cell respiration is carbs plus oxygen forms CO2, water, and ATP, right? This is how we're making energy in our body. You got to have oxygen. Well, if you're binding carbon monoxide instead of oxygen, you're not getting oxygen to your tissues. They can't make energy. And that's why people, when they are breathing in carbon monoxide, just go to sleep. They get really tired. Carbon monoxide is an odorless, colorless gas. So that's oxygen transport. Super easy. We just bind it to hemoglobin. Okay, now we do have some issues, but otherwise it's just bound to hemoglobin. CO2 transport is a little more complicated. We carry CO2 3 different ways in our blood, right? CO2 is a waste. We got to get rid of it. About 10% will just be dissolved in blood plasma. Why can't you dissolve more than 10% in blood plasma? When you add CO2 to a liquid, what do you form? Carbonic acid. And our blood pH is about 7.34, right? And if we change our blood pH, if we start adding acid to it, we're going to change our blood pH and we're not going to be able to perform a lot of metabolic functions. You can die from that. So this is why we can't carry more than 10% of CO2 in our blood plasma, because too much dissolved in plasma will start changing your blood pH, okay? 20% of CO2 is bound to the globin of your hemoglobin. Okay, so a little bit of CO2 can actually bind to hemoglobin. This typically happens in the tissues. When hemoglobin's already released oxygen, now it's free and open and it can take and carry some of that CO2, that waste. Okay, so this is usually happening down in the tissues. When carbon dioxide binds to the globin of hemoglobin, we call it carbaminohemoglobin. And so the Haldane effect just describes less oxygen you have bound to hemoglobin means you now have room for CO2. So again, this is why this is usually happening down in the tissues, not something that's happening in the lungs, okay? The remaining 70% gets transported as bicarbonate. So carbon dioxide will be converted into bicarbonate. Does that sound familiar, bicarbonate? Does anyone know what bicarbonate is? Bicarbonate is a buffer. So remember, we have talked about how your blood pH is so important. We have to maintain a blood pH of about 7.34, 7.35, right in there. When we convert CO2 into bicarbonate, it's acting as a blood buffer. So it's actually helping to maintain our blood pH. So the majority of CO2 gets transported as bicarbonate. So let's talk a little bit about the transport of CO2. So from your tissues into your bloodstream, we're pulling that waste. CO2 is going to combine with water and form, here's that acid, carbonic acid. We can't leave it like that. It will change our blood pH. So this is why we quickly convert it into bicarbonate. And this is happening in the red blood cells. So you can see the equation here. Here we've got our CO2 plus water. As soon as the CO2 moves into our bloodstream, it combines with water and forms carbonic acid. But we very quickly break a hydrogen off and turn it into bicarbonate. Okay, so we form that buffer. And bicarbonate is a great blood buffer. And how we get it to buffer our blood is we've got to put it in our blood plasma. It doesn't do us any good in the red blood cell. So as soon as bicarbonate is formed, it moves into the plasma so that it can help to buffer. But bicarbonate has a negative charge to it, so we have to balance the charges. If we're going to move bicarbonate out of a red blood cell, if it's got that negative charge, we're going to have to pump a negatively charged ion back into the red blood cell and balance charges. And what we put into the red blood cell is chloride ions. We call this the chloride shift. So let me show you a picture of this. So you can see here, this is our tissues. We can see our tissues. Again, they're going through cell respiration. They're using carbs and oxygen, and they're forming CO2, water, and ATP. So here's all the waste. Here's all this CO2 that they're making. We got to get rid of that from our tissues. So it's going to cross from our tissues into our bloodstream. Let me use a different color. We'll use black. There we go. It's going to cross from our tissues into our bloodstream. So you can see that CO2 moving into your bloodstream. And you can see here, CO2 combines with water, forms carbonic acid. It's an acid. We can't leave it like that. So it is immediately converted into, here's our bicarbonate. That's going to act as a blood buffer, but we got to get it out of this red blood cell and into the blood plasma. So you can see it's going to leave the red blood cell. Now it's in the plasma, and that's how we carry it. And it helps to buffer our blood. But because it has a negative charge, we have to balance our charges. So we shift some chloride into the red blood cell. Okay, and that's called the chloride shift. And this is how we carry about 70% of our CO2. Okay, so the majority of CO2 gets carried as bicarbonate ions. Now, once it gets into the alveoli, so once we've got that CO2 all the way up into the alveoli where we have to blow it out of the body, the reaction basically is going to happen in reverse. So here's that bicarbonate. 70% of the CO2 has been carried that way. So once it gets up into the lungs, we're going to add hydrogen to it and turn it into carbonic acid. From there, we're going to separate the water out of it, turn it back into CO2, move it into the alveoli, and blow it out of the body. So the reaction just goes backwards. It goes in reverse when we get it up towards the lungs, okay, near the alveoli. And the chloride shift happens in reverse. So here's the same picture, but you can see the arrows are now different. You can see we're up near the alveoli. We've got all of that bicarbonate in the plasma that's been doing a great job buffering. Now we got to put it back into our red blood cell. We're going to shift our chloride back out. So we're doing everything backwards. Here's our bicarbonate now. That's going to get converted into carbonic acid. We're going to disassociate the water from it. Now we've got our CO2 back in original form. We're going to move it across the respiratory membrane into the alveoli, and we are going to blow it out of the body. Okay. So I've already mentioned this buffer system a little bit. I want to talk to you a little bit about how buffers work. I don't know, how many of you have had chemistry? Anybody in here taking a chemistry class? You probably know how buffers work. Let me pull up a whiteboard. The way I think of blood buffers is I think of a blood buffer as like a hydrogen ion sponge, okay? So let's think of our pH scale. The pH scale runs from 0 to 14. 7 in the middle is neutral, okay? Now, if something is an acid, that means it is below 7 on the pH scale, has a low pH. Something's a base, it is higher than 7 on the pH scale. Okay, now, one thing to keep in mind is that because the pH scale only runs from 0 to 14, a change in one number is huge in terms of the change in acid content, okay? Now, in reality, when we talk about the pH scale, we're really talking about hydrogen ions. An acid has a lot more hydrogen ions in it. A base has a lot less hydrogen ions. So I think of blood buffers as like a hydrogen ion sponge. Okay, I want you to think of a sponge. A sponge can soak up extra water, right? Or you can take a sponge and squeeze it out. So it can either soak up excess or squeeze out if you need more. So I think of buffers as hydrogen ion sponges. So let's say, for example, let's do a different color. Let's say, for example, that your blood pH starts to rise. It starts to go up. As it goes up, you have less hydrogens. So a buffer would act like a sponge and squeeze the hydrogens out. Add more hydrogens into solution to bring you back down where you should be at neutral. If your pH starts to drop and become more acidic, That means you have too many hydrogens. And a blood buffer acts like a sponge and it soaks up the extra hydrogens, soaks it up, gets it out of solution to bring you back to where you should be. So blood buffers are either adding in the extra hydrogens if you need them, if you become too basic, or soaking up the hydrogens if you have too many. Okay, so it's going to either add extra or soak them up, depending on what you need. So that's why I think of a buffer as like a hydrogen ion sponge. Okay. All right. So the carbonic acid bicarbonate buffer system is just, like I said, it's acting like that hydrogen ion sponge. If hydrogen concentration in your blood goes up, that means it's becoming more acidic and it'll just soak up the extra hydrogens. If hydrogen ion concentration drops, that means it's becoming more basic and it will release extra hydrogens. Now, CO2 can influence, other than pH, it can also influence some other things within your respiratory system, particularly the rate and the depth of breathing. We also know that the rate and depth of your breathing can definitely affect your blood pH because it's influencing the amount of CO2 that's left in your body. So when you take very slow, shallow breaths, okay, so slow, shallow, like slow and like you're barely breathing at all, very slow breaths, very shallow breaths, you're going to start to increase the amount of CO2 that's in your bloodstream, meaning you're not getting it out. You're not blowing it out enough. So CO2 levels in your blood start to go up. And remember, CO2 is an acid. So the more CO2 you have, the more acidic your blood's going to start to become. So it's going to drop your blood pH. Rapid, deep breathing, like when someone is hyperventilating, right? That's going to cause CO2 levels in your blood to drop. Okay, they're going to go down. And that's going to cause your pH to start to go up and become more basic. This is why when someone is hyperventilating, big, deep breaths, maybe they just got bad news, they're freaking out, they're hyperventilating. What do you tell them to do? Breathe into a bag so that they can rebreathe CO2, a paper bag. Don't give them a plastic bag. Paper bag so that they rebreathe CO2 so they can bring their pH back down. Okay. Or you tell them, sit down and put your head between your knees. By doing that, you cannot expand your thoracic cavity as much, so you can't take as big of breaths in. It's naturally going to cause your breaths to become a little more shallow. Okay. So that'll help to equal everything out. So changes in ventilation play a huge role in the acid-base balance of your blood. Now, things that can control respiration, your brain, higher brain centers will control your breathing rate. Chemical receptors, we call these chemoreceptors. These are picking up pH changes. They're going to control your breathing rate, right? We just talked about how breathing rate can affect pH. So if your pH has changed, your body can alter your breathing rate to adjust. And then even reflexes. Okay, so neural controls. This is your brain controlling your breathing rate. Your medulla oblongata is basically your respiratory control center. Remember, the medulla oblongata is part of the brain stem. It regulates your breathing rhythm. And normal breathing is called eupnea. This means true breath. So this is just a normal breathing rate, about 12 to 15 breaths per minute. Also, your pons, which is also within the brainstem. This helps to also influence and modify your breathing rate. during speech, sleep, and exercise. The pons also helps to smooth the transitions between inhales and exhales. Also, there are some chemical factors that can influence breathing rate. CO2 is the most potent, most closely controlled. So your body, what affects breathing rate the most is CO2 levels. CO2 is a waste. You've got to get rid of it. It's not oxygen levels. It's CO2 levels that control your breathing rate. When CO2 levels start to go up, okay, that's called hypercapnia. When CO2 starts to go up, CO2, remember, when it's dissolved in a liquid, it is an acid, forms that carbonic acid. That's going to pass through the blood-brain barrier. It's releasing the acid, the hydrogen ions, and that's telling the brainstem, oh, we have too much acid, we have too much CO2, we need to increase our breathing rate. So this is actually going to cause you to increase the depth and rate of breathing so that you can help to get out more CO2. And that's going to help to regulate your pH levels. Now, I mentioned hyperventilation. Hyperventilation is when you breathe really deep and really fast, and it exceeds your body's need to remove CO2. This is going to decrease your blood CO2 levels. So you're blowing out way more than you need to. So CO2 levels start to drop. This can actually lead to things like dizziness and fainting. It actually can cause the vessels in your brain to start to constrict. It can cause what's called cerebral ischemia. That's what leads to the fainting. And sometimes if CO2 levels become incredibly low, it can lead to apnea, which is breathing cessation. Now, you've probably heard of sleep apnea before. Sleep apnea a lot of times will happen if somebody snores, or sometimes they'll stop breathing while they sleep. Like my husband has sleep apnea, and I can hear him breathing or snoring, and then suddenly I hear nothing. And then all of a sudden he'll do the really big snore, you know? And so apnea is when you stop breathing. Now, when CO2 levels start to get critical, okay, if you're not breathing, CO2 levels start to go up and up and up and up and up. And eventually it gets critical. You have way too much CO2. Your brain is going to go breathe, and you're going to go and take that big breath in. This is also what happens like when someone passes out, or if they faint, or if they stop breathing. The minute they pass out, first thing their body does, big breath in. This happens when people drown too. If someone's under the water, they're holding their breath, right? And eventually what's going to happen is when they're holding their breath, CO2 levels inside go up, 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 up. They're going to pass out. And the first thing they're going to do is take a big breath in. And if they do that when they're underwater, they're breathing in water. And so this is how they would know if someone has drowned, because when they examine them, if they can find water in the lungs, they know that they have breathed that water in, right? Now, the last thing that will control respiration are some reflexes. Okay, there are pulmonary irritant reflexes. You do have some receptors in your bronchioles that will respond to irritants. So if you've breathed something in that's aggravating, you've got to get it out. And the reflex for that is a cough or a sneeze. A cough is going to help you get out something that moved a little lower in the respiratory tract. A sneeze is going to help you get out something that's still up in the nasal cavity. An inflation reflex, you actually have some little stretch receptors in the pleura and your airways that are stimulated by lung inflation. And this is what is ending your inhale and your exhale. And so this is what's triggering that inhale and exhale. These are just some normal inflation stretch receptors that you have. They also act as protective response more than a normal regulating your breathing, in that when you try to take a big breath in and you keep breathing in and in and in and in, eventually your body goes, there's no more air that can come in here, and you blow out. That's the inflation reflex. It's a protective mechanism that says no more, you're going to do damage if you breathe more. right? So this is more of a protective reflex than anything. The last two are some adjustments that are made to our breathing rate. So exercise, this obviously will adjust our breathing rate. So hyperpnea is the term we use for increased ventilation. This is in response to metabolic needs, right? If you're exercising, you need more energy, which means you need more oxygen. However, when you're exercising, the CO2, oxygen, and pH remain constant because all that you're doing is your body is adjusting to your body's demands. So this is not like hyperventilating. You're not changing your oxygen or CO2 levels. Instead, you are maintaining those and you are just matching your body's demands by breathing faster. Okay, you're increasing your blood flow. One thing I'll point out is anaerobic respiration. This typically is not from poor respiratory function, but instead it's typically from insufficient cardiac output. So maybe cardiovascular wise, you're not putting out enough blood. So this is why it's really important to do cardiovascular activities to increase your ability to get that blood out where it needs to go. Also, sometimes your skeletal muscle is not able to take in that oxygen. A lot of times if you work out and you're not used to working out, when you do that, your muscles will swell and start pinching on your own blood supply. They actually cut off their own oxygen supply. This is when they go through anaerobic respiration. You feel real sore later. It's really common when you do weightlifting. Weightlifting activities will cause your muscles to swell, pinch on that blood supply, and lead to anaerobic respiration. So it's not typically from poor respiratory function. It's more from skeletal muscles' inability to get that oxygen or insufficient cardiac output. And then the last one is when you travel to a high altitude, you might get something called acute mountain sickness, AMS for short. This is when the atmospheric pressure and the oxygen levels are lower. So as you climb in altitude, the air gets thinner, oxygen becomes less. And this is when you get headache, shortness of breath, nausea, you might throw up. That's acute mountain sickness. Okay. Now the last couple things I'll let y'all do, these are all imbalances. So the last couple of slides from 22 all relate to some imbalances with the respiratory system. So you can read through some of those things like COPD and emphysema, bronchitis, tuberculosis, and lung cancer and cystic fibrosis. Okay. And then a little bit on the development of the lungs, which we've already touched on, especially with those type 2 cells. Okay. Anybody have any questions from 22?